The cell cycle is a four-stage process consisting of G1, synthesis, G2, and mitosis. G1, S, and G2 phases all occur in interphase. In the G1 phase, the cell increases in size and the cellular contents are duplicated, except the chromosomes. In the S phase, the DNA replication or synthesis occurs, meaning each of the 46 chromosomes is replicated by the cell. In the G2 phase, the cell prepares for the cell division by checking the duplicated chromosomes for errors and making corrections if needed, which is known as error correction. The M phase is mitosis, followed by cytokinesis, causing the formation of two identical daughter cells. The cell starts out in interphase, where DNA begins to condense into chromosomes. In prophase, the chromosomes condense and spindle fibers form. The nuclear membrane and nucleolus break down. Each copy of the chromosome is called a sister chromatid, and these are joined together by a single centromere. In metaphase, the chromosomes line up across the metaphase plate or equator of the cell and attach to spindle fibers by the centromere. In anaphase, the sister chromatids separate to opposite poles of the cell due to the spindle fibers retracting, assisted by the centromere. In telophase, the daughter chromosomes gather at the opposite poles of the cell. The spindle fiber breaks down and the nuclear member reforms. Cytokinesis is different in animal and plant cells. In plant cells, a cell plate forms in between the two nuclei. Cellulose is then deposited on each side to create a new cell wall. In animals, the cytoplasm constricts in the middle of the cell, creating a cleavage furrow, causing it to simply pinch off. In multicellular organisms, like humans, which are made up of organelles, mitosis only occurs in stemmatic cells. Mitosis is important for the repair of damaged tissue and the replacement of worn-out cells. It is also important for genetic stability by ensuring the precise and equal distribution of chromosomes to each daughter nucleus. The process of meiosis. The cell starts out in meiosis 1, prophase 1. In prophase, the nuclear membrane breaks down, chromosomes split into chromatids, and crossing over or homologous recombination occurs. Telomeres ensure that chromosomes do not stick to each other whilst also protecting genetic information. Crossing over or synapsis is when the arms of a pair of homologous chromosomes wrap around each other. The points at which they meet are called chiasmata. The arms break where they meet, exchanging genetic material between paternal and maternal chromosomes, which introduces genetic variation. Crossing over can put new alleles together in combination on the same chromosome. In metaphase 1, the chromosomes align in pairs called tetrads in the middle of the cell along the metaphase plate. Each pair lines up independently of the next. This process is called independent assortment. In anaphase 1, the chromosome tetrads separate and each chromosome moves to the opposite end of the cell. This is done through the spindle fibers attaching to the kinetochore. In telophase 1 and cytokinesis 1, two daughter cells form which are different to each other, and the chromosome number is halved. In meiosis 2, prophase 2, the centrioles duplicate and move to opposite poles. In metaphase 2, the chromosomes align at the equator of each cell along the metaphase plate. In anaphase 2, the daughter chromosomes move apart to opposite poles. In telophase 2 and cytokinesis 2, the cells divide once again, resulting in 4 daughter cells that are genetically different to each other and half the original chromosome number. In multicellular organisms, meiosis is restricted to the germline cells. Germ cells differentiate to produce male and female gametes, sperm and eggs or oocytes. Spermatocytes come from an immature germ cell called spermatogonia, which then differentiate to become spermatids, which then later become sperm. Polar bodies are byproducts of meiosis and have no biological role in fertilization or in embryogenesis. An oocyte is produced in the ovary during female gametogenesis. During oogenesis, the oogonia become primary oocytes. Meiosis ensures that the chromosome number of each species is maintained during sexual reproduction while also introducing genetic variation through crossing over and independent assortment. Malaria resistance is caused by a hemoglobin mutation called HBS, which causes the red blood cells to become curved. In 2001, researchers found that people with one copy of this gene are 29% less likely to get malaria, but people with two copies are 93% less likely.